occurring every second in every cell. And we're now the data on cancer. Basically, we're coming back to a concept we knew 50 years ago. You could irradiate and cause cancer very easily. In fact, when I was at Brookhaven, one of the main studies was on cancer of the thymus gland, because in the 40s, 30s, and 40s, they discovered that you could irradiate thyroid and control hyperthyroidism, but the thymus gland right next to it developed mutations that led to thymus cancer. And so by about 55, suddenly they realized, boy, you'd better not irradiate the thyroid. And so they came around to using iodine-131 that produces a soft beta and does the same thing. But we knew by the end of the 50s that the mutation rate, well, look at my arm, from ultraviolet.
Okay.
And then all of the ionizing radiations that I worked with all of them with corn in part.
So are you feeling health impacts? Are you feeling health impacts from that?
Am I feeling that?
Like, is your health affected by that?
Oh, no, well, I'm sure it is in some ways, but one of the things we've come to realize that my body is more bacteria than human.
Most people's are.
Something like 10 times as many bacterial cells, and they're mutating all the time. And in fact, one of my first courses at Cornell in '48 was on bacterial transmission of viruses. They call it bacteriophage.
Oh, bacteriophage, yes, okay.
And now we realize that many of the bacteria we, in our system and plant systems, are providing a home for viruses, and they in turn can mutate, as one of my main problems in the cornfield today is a virus that probably turned up in Peru about 40 years ago, new, and now finds its way around the world. Well, we're off the subject.
Can you tell me about the Ringspot virus while we're talking about viruses?
No, I really can't.
Don't know much about it?
Well, I'm not like an expert.
Gotcha. You're the corn guy.
Well, the resistance to the ring spot was classic stuff, and now the development of virus resistance, usually we just go somewhere and borrow it. In the case of corn, we have about 8 major viruses. I have 2 of them that I keep cooking all the time. I want them to be here so that all of my material is resistant. One is called maize mosaic virus. In fact, the day I arrived here, I got off the plane. Dr. Gilbert took me to a home that he'd rented for me. And the next day he brought me here to Waimanalo and showed me a virus, a cornfield full of virus, maize mosaic virus. And one of my first students found that the only place you could find resistance was in the Caribbean. So we went to the Caribbean and borrowed the gene, and that gene, chromosome 3, position 68, I have put into everything. I have about 2,000 types of corn that I work with. They're in a freezer out here, and they're part of a collection we call the Hawaii Foundation seeds that I started up in the 70s. After we started this Crop Improvement Association, we realized that one of the important differences when you move from Iowa to Hawaii, you're basically in two different corns. The corn of Iowa and the corn of the tropics are just as different as elephants are from kangaroos.
Right.
Corn, of course, evolved in the tropics. And you go back 5,000 years and it looked like this. Some of the earliest corn cobs are, of course, from southeastern Mexico. And I've long been an enthusiast, incidentally, of the Mesoamerican civilizations, the Maya. I wrote a paper on the Maya in '79 saying that the Maya collapsed because of maize mosaic virus, which I argue, and now some people are realizing I might be right, that among other problems that the Maya might have had, they were so dependent on corn, 50% of their calories. And you know, you go into the tropics today, most people are picking up 50% of their energy every day from either corn or rice or cassava. Now, cassava has become very big because it's the cheapest, but not the most nutritious.
Right. Humans have been modifying their food to be more nutritious for a very long time.
Yeah, indeed so. And now, of course, the tools of chemistry make that totally fascinating.
Yeah.
I would say for the modern plant breeder, nothing's more exciting than the potential improvements of the health of our foods. We've selected historically for calories, for energy, and just keeping us alive with the appropriate notable vitamins like C. Now we're realizing, especially in our Western world, where food is the cheapest thing we have, and we don't respect it.
Right.
Embarrassing to us in agriculture is that most people just don't realize how cheap their food is. They complain if they spend 10 cents more on something, and yet you go to India where people spend 50% of their money on food, or the Philippines, and we spend 13%, half of which we spend in a restaurant.
Right.
Where we have no idea how it grows.
We're so elite, it's such an elitism that comes out of being well-fed and having opinions on how the rest of the world should eat.
I've had the great pleasure, working from Hawaii, of course my focus is on tropical agriculture, and my love is, well, my first wife, second wife, were Filipina ladies. I went to the Philippines, my first big job after a postdoc in Sweden. I went directly by boat to the Philippines and worked 2 years in the early 50s. And I became enamored of not only the people and the foods of the tropics, but deeply committed. I'm undergirded by congregational Presbyterian philosophies with a motivation to see if I can do something about the terrible conditions of most people in the tropics. Even today, it's just hard for most people to recognize 1 billion people go to bed hungry, and nobody goes to bed hungry. We talk about poor people who don't have enough to eat, but they're not hungry. The way I lived in India, I've lived in Nigeria, I've lived long in the Philippines, Thailand, and when you see people that are really hungry. One of my last consultancies was up in northern north of Calcutta, up in this beautiful tea country. And we just don't have any understanding of how terrible foods are for most people in the tropics. Out of 7 billion. You know, when I was born, there were 2 billion people. Today there's 7 plus. And everything in agriculture relates directly to that.
Directly.
If we'd held 2 billion, we'd all be eating like you and I are today. When I went to the Philippines, there were 13 million, 1-3. There are now 100 plus million.
How do we keep up?
Well, if there were simple solutions, of course, they would have been gone through long ago. They are very profound. I don't pretend to be smart enough. I know that I firmly believe that we have lessons from our religion. We have lessons from democracy and conservative democracy that believes that we must educate and we must study and we must work and try to make a better world. And if you put all of those together, sometimes we have an impact.
Why are people so afraid of this technology, of transgenic genetic modification? Why are people so afraid?
I wish there were a simple answer to it. Whenever I sit down and grapple with this question, I realize that I just can't think except as a scientist. And as a scientist, I think, what's this all about? I've been modifying, when I have field days here, I say, every corn plant in this field has been genetically modified by me. I've brought genes in from here and there. I've struggled to find out that they are genes and where they are and how I can move them. And sometimes it just isn't one little old gene. The virus I work with now, I'm guessing 2 major ones, one of them from Peru. With other diseases, I just published a paper on tassels at the top of the corn. And a fascinating paper in Crop Science, which are our major publication. I had the great pleasure of being a founder of Crop Science 50-odd years ago. We have a Crop Science Association that is linked with a Soil Science Association in the Agronomy Society of America. And we have, from the Crop Science, published many reviews of evidence concerning the GMOs, of the use of transgenics. And none of the transgenes that have been approved have any material effect apart from that they were chosen for.
Can I ask a question? How, and this is always what people say, is how do you know? And you sound like you know, but how do you know that? Like, what's the process?
Well, immediately, of course, the process with any gene. Let's say I put this gene into corn.
Okay.
You probably wouldn't want it, but it's one gene. It's called double cob. And how do I find out whether that changes the chemistry of double cob, of the chemistry of the food, say the vitamin A? Because we're now tremendous amount of research on vitamin A or beta carotenoids with macular degeneration.
Golden rice.
Can you believe the figure from FAO? 500,000 children go blind every year in Africa. And that blindness is entirely due to the fact they eat white corn or white rice or cassava with no beta-carotenoids. We know those solutions and we can tell them that. But just like the GMO, you have ignorance initially to deal with. But come back to your question. The first thing, of course, with a GMO that Monsanto or anybody else has to deal with is to prove that there are no related effects other than that for which it was chosen.
Do you need water?
Yes, I do.
Karen?
Vivian?
Vivian's got it? Okay.
So, for example, I think one of the most exciting family of transgenes is the BTs that have been derived from Bacillus thuringiensis. Now, this fungus of the soil produces many chemicals that, thank you. And we've known long ago that among those chemicals are pesticides, or basically insecticides. And now a great family, I think something like 70 different BTs. Some will kill flies. Most of them kill insects that are chewing, like the corn borer. Well, the research to prove that there's no effect on anything else is classical. We do it all the time. You set up a series of corns that have and those that do not have this new gene, like this. They have to be isogenic, so which simply means we spend a lot of time creating those lines that are perfectly identical. And the fact is, this is perfectly identical, let's say, to this, except for one gene.
One gene.
This has gene tunicate, and this has one gene double cob. I, in 50 years of breeding, have put this gene into an inbred called Hawaii 27 from Colombia. I got it when I was in India 60 years ago, almost. Hawaii 27 I decided to use as the parent for the breeding of mutants like this. So this would be more typical Hawaii 27 with a white kernel and a black point on the top that we call Navajo. And then these are related ones that have genes affecting color, and some of them here affecting sugar, and this one affecting the cob. Those isogenic lines then are, in the modern context, put into a critical comparison, fed to animals, for example, typically fed to rats. I've done some of that myself. And while I'm not directly involved in that research, I know that by the time the Food and Drug Administration will approve a new transgene, those data would fill volumes, would just fill volumes. If I take my own research to prove that this is a monogene, it'd fill a pile like this. The techniques have to be spelled out precisely. So with the primary transgenes, and as you realize, there really aren't very many. The fact is, we're transferring genes like this, no need to create those genes. They're already there. Corn has roughly 35,000 genes, about much like humans, and they all mutate. And sooner or later, this would have been a normal gene that mutated into this strange bifurcation, that a cob that would normally be like this.
Right.
It's a poor example for me to be showing to you.
But so when you're doing a transgenic process, do you ever equate it to anything, or do you have an analogy that you tell people simply? It's like doing this.
Well, go back to what I'm doing, which is transferring genes that have not been created by me. They've been created by the Lord above as natural mutations. And a point that we all agree on is that sooner or later there would be a mutation that would make this Roundup resistant.
Right.
I mean, just wait long enough. Just wait long enough. I have an anticipation, and in a lot of cases, we look long enough, we find that those mutants are already here. A lot of people don't quite realize that the germplasm of any crop is so profound compared to what's on the shelf. You go down by an ear of corn and you think, well, corn is corn is corn, corn. But I've grown essentially because they're happy in Hawaii. Essentially all of the corns of the world are happy here. They come from Guelph, they come from Peru, they're equally happy. And we always find new mutants, just always find new mutants. We go out and suddenly here's a yellow ear with a purple kernel. Now, many of those, about 2,000 of them, have been plotted, and we carefully have found where they were. But the other 30,000 genes are mutating in ways very often we can't see, we can't identify. So the transgene specifically is unusual because you're transferring it from one organism to another. Increasingly, those transfers are going to be something that we can achieve normally. For example, if I find a gene in sorghum or sugarcane that I think, wow, it makes a plant perennial, it'll just go, well, I could bring that gene now into corn, because it's become possible with clever techniques to cross sorghum with something and then cross that to corn and bring the gene indirectly.
Wow.
And the same with sugarcane. And it turns out corn, sorghum, and sugarcane are distant relatives, but they're all in the Anthropogoniae in a little family that belong together, and you begin to look at them and you say, well, yeah, I see they are.
So, I mean, knowledge it's a progression, and it sounds like through the knowledge of transgenics, we have further refined that efficiency. That's a very interesting idea that you just described.
Well, there's no question in my mind, increasingly, we're going to exploit this. A lot of that we've become clever at growing from a cell. You hear about stem cells, And now we're talking about taking a stem cell from me, giving it to you, because I'm resistant to measles or something. Believe me, 50 years from now, that'll be as common as me crossing this with this. I mean, quite seriously, the evidence that human diseases can be controlled by moving genes, by giving the cells that carry those genes is very real. We look at inherited diseases or diseases with a strong genetic component, like diabetes. Is it the type A that children often early in life have? Well, it turns out most of the people in the tropics are resistant, and a high proportion of people in northern Canada and northern Europe are susceptible, the frequency of that disease. Now there's very strong evidence that this can be controlled, and I'm getting away from something that I can't give you the facts on.
Okay.
But I'm getting now, I'm very impressed. Almost weekly we hear about gene jockeying in the form of moving a cell. And not necessarily creating the new gene. But let's come back to the created transgene. The fact is now we know the structure of DNA down to the last digit in many, many organisms. Corn isn't quite completely mapped, but the human genome essentially completely mapped. So increasingly our biotechnologists can talk about a specific segment of a specific chromosome that appears to have specific capability, which now you can extract, classically put it into Escherichia coli, the common colon bacillus, and use that bacterium to transfer it to another organism. That technology, as strange as it may seem, I think already nature has been doing that for years. We come back to my model of the bacteriophage, where now we're realizing, you shake my hand, we share many, many bacteria now. If some of those move through the system, they could presumably transfer genetic material. But coming to the more practical features, we, of course, we'd like to improve both the yield and the quality of many of the foods of the world. And when you consider that by giving corn one gene for resistance to corn borer, has, in a country like the Philippines, has doubled the yield. I mean, if you'd go out into my cornfield now and tell me I can get 2 ears for the price of one that I had last year, no way. Well, that's exactly now what has happened. So here in the United States, 95% of our corn carries the gene BT.
Right.
95% of our cotton carries the gene BT. I was seeing just this week the BT cotton story. And when I lived in Thailand in '67, 26 farmers died from the use of parathione controlling cotton seed weevils and cotton insects. Today, the yield of cotton in Thailand is up 400%. No parathion, and no farmers dying. The reason I knew that, one of the farmers who worked with me on corn lost his own child because the child got into the parathion, not into the cotton field.
Right.
And so we are seeing a dramatic impact on the yield of crops like this. Now, there are a lot more important ones coming along if people will permit them. A perfect and interesting example, coming back to this concern about beta carotenoids and blindness, that is immensely popular. It's hard for us in America to realize how common. When I was working in Australia a few years ago, they got a large grant to make sweet corn with high beta carotenoids, and I've been working with them because what we're looking for is something like this, very bright.
Orange, yellow, golden.
Yeah, and you go into this, you find that the carotenoids break off with an alpha series and a beta series, and the beta series ends up with a thing called zeaxanthin. Zea is corn.
Yeah.
And beta carotene and zeaxanthin, beta carotenoids are important as provitamin A, but the zeaxanthin directly affects the eye development. So you find people who are regularly taking an ointment of zeaxanthin and lutein for this problem. But it is really amazing when you go into primitive parts of the tropics to see how many people are blind, or if not blind, at least having great difficulty seeing, and not something that you can do by putting in a new lens.
Does it frustrate you when we have solutions to food problems?
Of course. I would be very happy with yellow rice.
Golden rice.
No problem at all. But people around the world, white means clean, white means free. And silver, we talk about silver corn or silver rice. Getting people to eat yellow rice is going to be a big challenge, probably not easily, but it's available, and you know where they got the gene? From corn.
I met Dr. Ingo Patrikis, who did golden rice.
Yeah.
I got to shake his hand.
The golden rice at Erie at the Rice Institute in Los Baños, they jockeyed genes from several. I mean, you can find beta carotenoids in a lot of foods, obviously papayas. But they found the best gene to do was the gene on chromosome 6 in corn. At the end of chromosome 6 in corn is a simple gene, 2 alleles, one white, one yellow, and it simply breaks this anyway. Well, there are now 2 other genes I'm working with. One is, both of them are pale yellows. And a lot of the corn in Iowa is pale yellow. It isn't this rich yellow. So if I'm helping the Gates Foundation breed a corn, which they're testing in Zambia, the center of that research is Purdue. The Zambia corn is building on some of my material that has high provitamin A. And and they are in Zambia totally eating white corns, which actually the story of that is, I won't go back, but in the olden days, Africans ate yellow corn, but when, after World War II, the Europeans came in, and in East Africa, especially in the colonial, corn became one of the major products for the European market, and for things like polenta, they got to have white corn. So whites took over, and curiously, most of it came from southern United States, Hickory King, famous old corn, that white corn. But
could you tell me about, we've visited a few Monsanto sites in the last few days.
yes, oh good,
Yeah, I've seen some corn growing.
of course.
Can you tell me how those companies came to be in Hawaii and what the plan is there and what the idea is?
Well, a little bit of that. I wrote up those reminiscences.
Water as well if your throat is sore.
I'm always a little croaky.
Oh, you probably talk a lot, don't you?
Not like I used to. I used to lecture regularly, but I'm on the research faculty, but I love to teach, so I did teach. I came here in 1961, and mind you, I had been working in the Brookhaven laboratory that I talked about. So I came along excited about radiation mutation and studies of basic biotechnology, what we call today. But I was introduced immediately to this problem of virus. Sweet corn used to be a major product in this area, but the virus had driven them out. And Dr. Gilbert, the head of the department, said, well, here's a problem for you. I said, I haven't been working much on corn, although when I was in the Philippines in the early 50s, they put me on the RICO project, rice and corn, the RICO project. And we worked on both rice and corn. So, and I grew up with it. My dad got his PhD on corn, so long before. I helped pollinate when I was a kid. Anyway, I picked up immediately with my students the sweet corn development for Hawaii. And we were, of course, new to the islands and wanted to get acquainted with every place. So I planned experiments in a classical way. Well, I need to have it on this island, and this elevation, and this elevation. So by 1964 or so, I had 8 different research sites, and I was often hopping on the plane, $12 a trip back in those days, to go to Molokai or whatnot, and have these experiments collaboratively with growers, which was good fun. And indeed, we pretty quickly came up with a new corn we called Hawaiian sugar that everybody liked. So, but by a major series of those was on the island of Molokai. And it turned out that Molokai had never had sugar cane. And now in the other islands, sugar cane basically took up all the good soil.
Right.
So Kauai, Oahu, Maui, the good soil was gone. So anybody who thought about big production of corn was in trouble. But we found that Molokai, the sugarcane people had come a little before World War I and World War II and decided, no, they'll go to Kauai instead. They did come here for a while and they left. This whole region of Waimanalo was in cane. So there was no sugar cane, and the pineapple industry on Molokai was dying. So we saw it as a nice opportunity for corn production. But by then I was well aware of the fact that our mainland friends were working, accelerating the breeding program by having a second generation every year. They spoke of that as the winter nursery. And they tried going to, Pioneer went to Mexico and then Nicaragua. They tried going to Puerto Rico. And they set up in southern Florida, one of the major areas for this winter nursery. Well, having visited these places myself, I realized that Hawaii was much more congenial for several reasons. So I published a note around 63 or 4 with the seedsman summer meeting of the crop improvement that they ought to come to Hawaii. And 2 of them sent seeds over in 65. And they came over. I thought the seeds looked awful, but they were quite happy. In the winter, we were with the short days. The corn coming from the mainland is very unhappy. But these 2 were Illinois Foundation seed and Corn Nuts Incorporated. And they were excited by the luck we'd had with the 3 trials I planted for them. And the next summer, they come over and we went around the islands and decided to get started on Molokai. And Pioneer generously rented 5 acres for us. And so in the winter of 66, we had a whole 5 acres. And I took my nursery over there, too, to compare growth here. By that time, my own program, I'd figured, well, winter, summer, it's all pretty much the same. So I just planted when I wanted to plant. And indeed, over the years, I've averaged at least 2 to 3 plantings per year.
That's huge when we're talking about producing seed stock for the rest of the country.
If you've got, say, a hot new gene that you want to move, I didn't back up to standard gene movement. We like to backcross 6 times minimally. Pioneer would backcross 10. So 10 generations.
Whoa.
With a
10 years.
hot new gene. Let's say, oh, I've got a gene for purple corn. People want purple corn. Okay. To put it into my favorite parents, you're talking 10 generations. I get by with 6, but even with 6, there are still some genes there that I didn't want them to come in. Long story short, that now the technology has so improved because there's a technique called dihaploidization where you can circumvent that to some extent. So in 1966, 67, I spent in Thailand, 68, bit by bit, and Don Shaver decided to start what he called Molokai Seed Service. I had set up, incidentally, moving back, we created on the campus Seed Industry Council because I argued, along with my colleagues, wonderful place for seed production. And rather than think about broad area production, say of tomatoes and strawberries, Hawaii would be a much better place to concentrate on crops where you're producing the seed rather than the product. And while we've come a long way with corn, there are a number of other crops that fit that. I had, in fact, been working with petunias. I worked 5 years on petunia when I was in the atomic energy. It was an excellent crop for studying mutations. And the petunia industry moved to Costa Rica with its seed production. So that was part of my motivation. I'd been down to Costa Rica and said, well, if you can take petunias to Costa Rica, why don't you bring them to Hawaii? I never succeeded with that one.
So how is the current anti-GMO environment making it difficult to do your work here?
Well, it has no effect at all on my work because I am not directly involved in transferring genes that have been transgenic type. I have the great virtue in corn of basically finding any gene. And partly the reason that's so is because any corn likes Hawaii. Now, if I were up in Guelph, a lot of the corn of the world I simply couldn't grow. But I realized right away that was different here. I add to my story. In the summer of '48, I'd finished my bachelor's, and I hitchhiked out to Caltech and spent the summer at Caltech on their maize genetics program. Dr. Beadle, one of the Nobel laureates, headed that program, and they had recently brought back seeds from Anahuac and that had been under the radiation out in the Pacific, Bikini and Anahuac bombs. And boats set around those had corn samples, which came back to Pasadena and were growing in the cornfield. And that summer of '48, a number of scientists came, and I just had written and said, can I have a job? And they said, sure, come out and pollinate corn, which I did. And here was all this damned exotic corn that had been bombed, all kinds of strange mutants coming up in the cornfield. Every day we were finding something oddball new. So I got totally fascinated by the event of mutation and began to get on this track of trying to find the beneficial ones. And that turned out to be the real problem because most of them are deleterious, not beneficial. But anyway, that summer I saw all this exotic germplasm. And so when I came to Hawaii, the first thing I did was check, and there are 220 major races of corn, like Peru. I gathered seed of all of them, and they all grew very nicely here. So for years we had the stupidest looking cornfields. Ears that are like this, ears that are like this, ears that are flexible. And it's such a fascinating collection. And of course, primitive people loved specific kinds of corn. So one group of Indians would have black, and another group of Indians would have red. And they had priapic ceremonies where a red ear had connotations that I shouldn't get onto your press. Anyway, in this process, of course, we began to realize that there were still segregating in these. Because they're open pollinated races, they're like races of people. And you suddenly look at 1,000 or 10,000 plants in a way that you can't easily with cows or people. You're finding that there are mutations in every field.
So I want to ask you some questions about the group with knowledge and the group with fear. Because we had just spoken about that earlier.
Yes.
How do you unite those two groups?
You know, there are people could approach that much more delicately than I.
Yeah?
As a scholar and a teacher of science, my answer would generally be start with kids 3 years old and make sure that the science training is up to 2000, not 1900. About half the people in the United States do not believe in evolution. The data proving evolution, I believed it when I was a graduate student. There was no question that genetic evolution was occurring. And now, every day you see something about, well, corn evolved 4.8 million years ago, and it looked like this, but about 100,000 years ago, it began to look like this. Well, we have that kind of evidence as scientists, overwhelming. It does suggest then that 50% of the people in the United States today, and there aren't quite as many in other developed countries, have either philosophical attitudes, which can be part of it, or are simply ignorant of the science of evolution. Well, you add to that anything as complicated as the modification that occurs in genes, mutation alone, is studying that in grade school and high school has to be solidly founded. And there's no question, we're embarrassed to admit it, but Hawaii is one of the worst states for science education. Our grades are among the worst in the country, and the United States has one of the worst science levels of the developed countries. Don't compare us with Sweden or Denmark or New Zealand. Just don't, or Korea. Don't be unwise, because we are not motivating those studies. So I start with education as an educator and say, we must have that. But then beyond that, as I realize that people have fears that have to be addressed by people who understand fear, and I really don't. Science to me, there are answers to questions. So I fear problems with my own health and whatnot, but they're not like, I think that there's something strange that can't be studied and solved. Anya Fishorik on our faculty.
We talked to her yesterday.
Good. Well, now she's the kind of person who you can sit down and get a much more intelligent answer to that. I only know that there is, and she has, as I've spoken to her, she's confirmed that I simply don't know how to deal with the fear part of it.
I've got one more question for you.
Sure.
Okay. And this comes back to whatever people's religion is, whatever they call their universal creator.
Yeah.
The general sentiment is that to meddle with nature is to go against that creator. Yet I've never felt that, and I'm sure you have an answer to this. Mm-hmm. Do you feel that it's against God to be doing this work?
I wouldn't even think of that. Curious. But I do recognize there are people who feel that way. No, not at all. my God is very liberal as far as science is concerned. My God created the mutations I'm looking at, if you wish to, and most of them are deleterious. So my God isn't just always making things better. Indeed, evolution is full of failures, and the mutational process that we build on will be much the same. Certainly things like this won't go to the market. But no, I don't. I firmly believe, of course, in a higher I don't know just how to put what my belief is. I've been a founder of my church, and I've been a teacher, and I've spent many, many years on the session of the church, and yet I'm still learning what God has for me. When my heart started going out here a couple of years ago, I had to sit back and think. I don't really think that there's anything about this that you can accuse any higher power of meddling, or you're meddling with nature.
Thank you for that answer. That's a good answer. I think so. Karen, what did you want me to ask?
Just where he sees. He's seen so much history.
Yeah. Where's it going?
Yeah.
What do you
Look
see?
at it, yeah.
What does the next 20 years in food look like?
Well, there's no question in my mind that plant breeding is here to stay. Animal improvement, plant improvement, human improvement. They all will involve moving genes. They will all involve genetic modification. So the overall concept is healthy, wise, and the most important aspect is that we keep the eye on improving rather than going the other way, which I suppose could be possible in the old Hitlerian sense. But I have no question that there will be transgenes coming affecting all crops. Partly, this isn't because we can't find the genes. In most cases, if you look hard enough, you'll find that nature created them already. But to be able to patent them and make money off of them and then get them into broad use is the challenge. But there's no question that there are mutants that will certainly we'll be using bacteria and viruses to move genes among species in ways that won't necessitate the patenting process or the use of the big companies. But you can't look at a crop in the tropics that doesn't have a major family of problems that could potentially be solved often by just bringing a gene from another plant. We're looking at a very serious greening disease.
Citrus greening.
Of citrus. And when visiting a couple years ago, they showed us the transgenic planting of 2-3-year-old citrus, absolutely resistant, and that gene was jockeyed from lettuce, if I remember.
I think that's so cool.
I
mean,
think
I
it's cool.
understand why you're fascinated by it. It's neat.
Yeah.
It's just neat. Thank you for your time.
Yeah.
I mean, that's
Pleasure.
it. We're done. We're done.
Yeah, well, great.
Perfect.
I'm
Oh, thank
sorry
you.
my old voice here gets